Hello everyone, welcome to Biomix-Up. In this video, we are going to discuss about the Calvin cycle or C3 cycle. So before discussing about the Calvin cycle or C3 cycle, we have to know why this name has been given so. This name has been given because the first stable product which is formed in this cycle was a 3 carbon containing compound. That is the reason it is known as C3 cycle, and the scientist who given this cycle was Calvin. That is the reason it is also known as Calvin cycle. So before discussing about this, we have to know where the cycle is taking place. This cycle is taking place in the stroma of chloroplast. Okay, and for this cycle to take place, the first enzyme which we need is Rubisco. And this enzyme can work as oxygenase as well as carboxylase. Okay, depending upon the concentration of oxygen and carbon dioxide, it will work as oxygenase or carboxylase. If the concentration of oxygen is more and the temperature is high, it will work as oxygenase, and if the concentration of carbon dioxide is more and the temperature is minimum or optimum temperature is present, it will work as carboxylase. So what happens in this cycle? The carbon dioxide which is present in the atmosphere need to be fixed, and in this cycle the carbon dioxide is fixed. At a time only one carbon dioxide can be fixed, but for balancing the equation we will see here that 3 molecules are utilized at a time. Okay, so if the carbon dioxide is fixed, we are giving the carbon dioxide, so the plant need to utilize it, someone need to accept it. So the first acceptor of carbon dioxide is a 5 carbon containing compound which is RuBP, which is also known as rubulose bisphosphate, and it is a 5 carbon containing compound. And for this reaction to take place, for having a balanced equation, we are using here 3 molecules of RuBP. So when we are using 3 molecules of RuBP, we can fix 3 carbon at a time, but naturally only 1 carbon dioxide is fixed when we are doing a cycle. But for balancing this equation, we are utilizing here 3 carbon dioxide at a time. 3 carbon dioxide is fixed. So 3 carbon dioxide is utilized here. That means it is going to be fixed. So 3 molecules of RuBP is needed to accept this carbon dioxide. And for this reaction to take place, here the Rubisco enzyme is used, and this Rubisco enzyme will work as carboxylase over here. Okay, so we are using here 3 molecules of 5 carbon containing compound. That means we are utilizing 15 carbon. Okay, from here we are getting 3 carbon. From carbon dioxide we are getting only 1 carbon. So here whenever we are using 3 carbon dioxide, we will get 3 carbon. So now we will get next compound which is a 6 carbon containing compound, 5 from RuBP and 1 from carbon dioxide. We are getting 2 carboxy 3 keto arabinitol 1,5 bisphosphate. It is a 6 carbon containing compound. Okay, so here we were having 15 carbon from RuBP and 3 carbon from carbon dioxide. So we have here 18 carbon. 3 molecules of 6 carbon containing compound equals to 18 carbon. Okay, but this is a unstable compound. So for stabilizing compound we need H2O. Okay, here H2O is utilized to make this compound stable. The first stable compound is 3-phosphoglyceric acid. This is a 3 carbon containing compound. That is the reason this cycle is known as C3 cycle. As this is a 3 carbon containing compound, for balancing the equation we need here 6 molecules of 3 carbon containing compound, so that we will get here 18 carbon. Okay, now what we have? We have 3-phosphoglyceric acid. Now we have to add a phosphate group over here to form 1,3-bisphosphoglyceric acid. Here in 3-phosphoglyceric acid we have only 1 phosphate in the 3rd carbon, but we have to give another phosphate to form 1,3-bisphosphoglyceric acid, so that phosphate will be present in 1st number carbon as well as 3rd number carbon. So whenever we have to add a phosphate, we need a enzyme which is known as kinase, and before this kinase we have to add the name of the substrate, which is 3-phosphoglyceric acid. We can write 3-PGA also to 3-phosphoglyceric acid. So for this reaction we need a enzyme which is known as 3-phosphoglyceric acid kinase or 3-PGA kinase. Kinase is just an enzyme for adding the phosphate. It will help in adding the phosphate. But we need phosphate. From where we will get the phosphate? We will get the phosphate from ATP. Okay, we are using here 6 molecules. So we will use here 6 molecules of ATP, and in return we will get 6 ADP. 1 phosphate will be utilized over here to get 1,3-bisphosphoglyceric acid. This 1,3-bisphosphoglyceric acid is also a 3-carbon containing compound. Now we have got 1,3-bisphosphoglyceric acid. It is in the acidic phase now. This acidic phase will be converted into glyceraldehyde phase, and this step is known as reduction step. For this process to do, we need a enzyme that is known as NADP, glyceraldehyde 3-phosphate dehydrogenase. This is the enzyme which we are utilizing. It is known as NADP, glyceraldehyde 3-phosphate dehydrogenase. As we have told that it is a reduction phase, that means something is going to be reduced. 6-phosphate is being utilized over here, so 6-phosphate will be reduced in this phase. Okay, and 6-NADPH will be converted into 6-NADP plus. Okay, and we will get 3-phosphoglyceraldehyde. How many molecules of 3-phosphoglyceraldehyde? 6 molecules of phosphoglyceraldehyde, because it is a 3-carbon containing compound. So for balancing the equation, we need 6 molecules of 3-carbon containing compound, which in total gives us 18 carbon. So we are having here 6 molecules of 3-phosphoglyceraldehyde. 1 molecules of phosphoglyceraldehyde will be used. How it will be used? It will be used in the stroma for the synthesis of starch, and it will be used in the cytosol for the synthesis of sucrose. If we need starch, it will be present in the transported into the stroma, and if we need sucrose, we need the synthesis of sucrose, then it will be transported into the cytosol. So we have to know what is starch and what is sucrose. The starch is the stored form of glucose, and sucrose is the transported form of glucose. Okay, till now we have discussed the carboxylation phase and the reduction phase. Let us see again about the carboxylation phase and the reduction phase. What happens in the carboxylation phase? The carbon dioxide is being fixed in presence of the enzyme rubisco. Okay, when the carbon dioxide is utilized, it will be accepted by RUBP, which is a 5-carbon containing compound, and it will give a 6-carbon containing compound, which is known as 2-carboxy-3-keto-arabinitol 1,5-bisphosphate, and this compound is a unstable compound. For the stabilization, we need H2O, and in return we will get 3-phosphoglyceric acid, which is a 3-carbon containing compound. Okay, here we are getting 6 molecules of 3-carbon containing compound for equating the equation, so that we will get 18 carbon in return. 6 molecules of 3-carbon compound will get 18 carbon. Okay, now this 3-phosphoglyceric acid will be converted into 1,3-bisphosphoglyceric acid, and for this we need a enzyme that is 3-PGA kinase, and 6 ATP will be utilized over here, and we will get 6 ADP. Now this 1,3-bisphosphoglyceric acid is in the acidic form. Now it will be converted into aldehyde form. Okay, how it will be converted? By the reduction phase it will be converted. Now it will be reduced. For this it needs a enzyme that is known as NADP, glyceraldehyde 3-phosphate dehydrogenase. As it is being being reduced, 6-phosphate will be given out. First of all, 6-phosphate was utilized, now 6-phosphate will be given out, and the NADPH which was present, it will be converted into NADP plus, or it will be reduced into NADP plus for giving 3-phosphoglyceraldehyde. 6 molecules of 3-phosphoglyceraldehyde will be given. Okay, now we are having here 6 molecules of 3-phosphoglyceraldehyde. From here, 1,3-PGAL will be used in the stroma or cytosol for the synthesis of starch and sucrose. Now, we have discussed about the carboxylation phase and the reduction phase. Now comes the regeneration phase. The RUBP which is being utilized over here needs to be regenerated again so that it can fix another carbon dioxide. For this, we have to regenerate RUBP. Let us see what are the steps involved in the regeneration phase. So, what we have did here, we have utilized one molecule of 3-PGAL for the synthesis of starch or for the synthesis of sucrose. Now, what we will do, we have 5 molecules of 3-PGAL. Now, from this 5 molecules of 3-PGAL, 3 molecules of 3-PGAL will remain as it is. Okay, but what about the rest 2? The rest 2 will be converted into its isomer form. It will be converted into DHAP. DHAP is an isomer of 3-phosphoglyceraldehyde. For this, we need an enzyme which is known as isomerase. So, we are having here 2 molecules of DHAP and 3 molecules of 3-PGAL. From here, we will take 1 molecule of DHAP and 1 molecule of 3-PGAL. And in presence of the enzyme aldolase, it will be converted into fructose 1,6-bisphosphatase. This DHAP was a 3-carbon containing compound and this 3-PGAL was also a 3-carbon containing compound. So, in return we will get a 6-carbon containing compound which is known as fructose 1,6-bisphosphatase in presence of the enzyme aldolase. Okay. Now, what we will do in the next step? The fructose 1,6-bisphosphate is having phosphate in the first number carbon as well in the 6th number carbon. We have to remove one phosphate from here. From first number carbon, the phosphate will be removed. For this, we need an enzyme which is known as fructose 1,6-bisphosphatase. Whenever we are adding a phosphate, we need an enzyme kinase. Whenever we are removing a phosphate, we need an enzyme phosphatase. Okay, now we got here fructose 6-phosphate. Okay, now from here we have utilized from this 3-PGAL, we have utilized only one molecule of 3-PGAL. Now the remaining 2 are there are next 2 molecule of 3-PGAL. From that 2 we will utilize another 1 molecule of 3-PGAL. Okay, with fructose 6-phosphate. Now this fructose 6-phosphate and 3-PGAL in presence of the enzyme transketolase will give erythrose 4-phosphate and xylulose 5-phosphate. Okay, this erythrose 4-phosphate is a 4-carbon containing compound and this xylulose 5-phosphate is a 5-carbon containing compound. So we have here having total of 9-carbon containing compound in the split form. Okay, because we have utilized here 9 carbon, from fructose 6-phosphate we have utilized 6 carbon and from 3-PGAL we have utilized 3 carbon. Okay, so now this erythrose 4-phosphate will be combined with DHAP. Here we have utilized only one DHAP. There is another one DHAP present. So this DHAP and erythrose 4-phosphate in presence of the enzyme aldolase will give pseudoheptulose 1,7-bisphosphate. It is a 7-carbon containing compound. This DHAP was a 3-carbon containing compound and erythrose was a 4-carbon containing compound. So in total it is 7-carbon containing compound. In presence of the enzyme aldolase, it will give us a compound known as pseudoheptulose 1,7-bisphosphate, which is a 7 containing compound. Okay, So here we are having phosphate in the first number carbon and in the 7th number carbon. So we have to remove one phosphate from first number carbon. We will remove a phosphate. So for removing the phosphate we need a enzyme. The enzyme is phosphatase pseudohaptulose 1,7-bis phosphatase. Okay. So when we remove this phosphate, we will get pseudohaptulose 7 phosphate. Okay. Now we have utilized this 2-DHAP and we have utilized 2-PGAL, one in this step and another one in this step. So there is remaining of 1-PGAL. This 1-PGAL, 3-PGAL will in presence of the enzyme transketolase by combining with pseudohaptulose, it will give ribulose 5-phosphate and xylulose 5-phosphate. These are the 5 carbon containing compound. Ribulose 5-phosphate is a 5 carbon containing compound and xylulose 5-phosphate is also a 5 carbon containing compound. We have utilized here 3-PGAL, which was a 3 carbon containing compound, and pseudohaptulose was a 7 carbon containing compound. Okay, in presence of the enzyme transketolase, it gave ribose 5-phosphate and xylulose 5-phosphate. Now, this ribose 5-phosphate will be converted into its isomer form in presence of the enzyme isomerase into ribose 5-phosphate. Okay. Now, what about this xylulose 5-phosphate? Here we are having only one molecules of xylulose 5-phosphate, which is a 5 carbon containing compound. Okay. Here we are having erythrose 4-phosphate and xylulose 5-phosphate. This erythrose 4-phosphate has been utilized here, but this xylulose 5-phosphate is free here. It is not utilized. So from here one molecule of xylulose 5-phosphate is present and from here one molecule of xylulose 5-phosphate is present. So we are having here 2 molecules of xylulose 5-phosphate. Okay. So here we are having 2 molecules of xylulose 5-phosphate, which is a 5 carbon containing compound. So in total we have 10 carbon containing compound over here. This xylulose 5-phosphate will be converted into its epimer form, that is ribulose 5-phosphate in presence of the enzyme epimerase. Okay. The epimer form of xylulose 5-phosphate is ribulose 5-phosphate and the isomer form of ribose 5-phosphate is also ribulose 5-phosphate. Ribose 5-phosphate we have got 5 and from xylulose 5-phosphate we have got 10 number of carbons. So in total we have got 15 carbon. This ribose 5-phosphate is having 5 carbon, which is of one molecule, and xylulose 5-phosphate is also a 5 carbon containing compound. So it is having 2 molecules of xylulose 5-phosphate. So in total we got 3 molecules of 5 carbon containing compound, that is 15 carbon. Okay. So what we have to regenerate? We have to regenerate RuBP, which is also known as ribulose bisphosphate. So we have got here ribulose 5-phosphate, but we need ribulose bisphosphate because ribulose bisphosphate, which is also known as RuBP, it is also a 5 carbon containing compound and it is responsible for fixing the carbon or accepting the carbon dioxide for the fixation. So this ribulose 5-phosphate will be converted into ribulose bisphosphate. That means we here need one phosphate to be added. Whenever we need a phosphate to be added, we need a enzyme that is known as kinase. Which kinase? Ribulose 5-phosphate kinase. Before the kinase, we will add the substrate's name. Okay. So we need a enzyme kinase over here, plus a phosphate is also needed, which needs to be added. So here we need ATP. This ATP will be converted into ADP by donating one phosphate. How many molecules of ATP is required? As we are having here 3 molecules of this 5 carbon containing compound, we need 3 molecules of ATP. so that we will get RuBP. In case of RuBP, there is two phosphates present in the first position and in the fifth position. So we are getting here RuBP. Now the RuBP is again regenerated. This RuBP will help in accepting the carbon dioxide which need to be fixed, and the cycle will continue another time. Okay, so we have completed this. Let us have a quick revision of this slide. So what happens? There is a RuBP present and a carbon dioxide need to be fixed. RuBP is a 5 carbon containing compound. So from here we are getting 5 carbon, and from carbon dioxide we are getting 1 carbon. Here we are utilizing 3 molecules because for balancing the equation. But in nature there is fixation of only 1 carbon dioxide at a time. Okay, so from here we got 5 carbon, from here we got 1 carbon. So we are getting here a 6 carbon containing compound which is known as 2-carboxy 3-keto arabinitol 1,5-bisphosphate, and it is a unstable compound. For this reaction to be done, we need an enzyme that is known as Rubisco. This Rubisco can work as carboxylase as well as oxygenase depending upon the concentration of carbon dioxide or oxygen. Okay, as this is an unstable product, we need it to be stable, so that we need H2O. Then it will be converted into 3-phosphoglyceric acid, which is a 3 carbon containing compound, and this is the stable form. So for balancing the equation, we need 18 carbon in total. As this is a 3 carbon containing compound, we need 6 molecules of 3-phosphoglyceric acid to give 18 carbon. The 3-phosphoglyceric acid is converted into 1,3-bisphosphoglyceric acid in presence of the enzyme 3-phosphoglyceric acid kinase, and by utilizing 6 ATP and converting it into 6 ADP. This is known as carboxylation phase. Next comes the reduction phase. In reduction phase, this 1,3-bisphosphoglyceric acid is converted into its aldehyde form. This is the acid form. It will be converted into the aldehyde form. So for this, it needs an enzyme NADP glyceraldehyde 3-phosphate dehydrogenase. As it is a reduction reaction, 6-phosphate will be removed and 6-NADPH will be converted into 6-NADP plus, or we can also say it that 6-NADPH is reduced into 6-NADP plus. Now we have got here 3-phosphoglyceraldehyde or 3-PGAL. Of it, 1 molecules of 3-PGAL will be converted into starch or sucrose. If it is secreting the starch, it will be given to the stroma, and if it need to synthesize sucrose, it will given into cytosol. Next, remaining 5 molecules of 3-PGAL. Of this 5 molecules of 3-PGAL, 3-PGAL will remain as it is, and 2 molecules of it will be converted into its isomer form, which is DHAP. Now from this, 1 DHAP and 1 PGL will be converted into fructose 1,6-bisphosphate with in presence of the enzyme aldolase. In the next step, what happens? This 1,6-bisphosphate will be converted into fructose 6-phosphate in presence of the enzyme fructose 1,6-bisphosphatase. 1 phosphate will be removed over here. Next what happens? There is another PGAL. This next 1 mole of 3-PGAL will be converted into erythrose 4-phosphate and xylulose 5-phosphate in presence of the enzyme transketolase. It will combine with fructose 6-phosphate and 3-PGAL, and it will be converted into erythrose 4-phosphate and xylulose 5-phosphate in presence of the enzyme transketolase. Now this erythrose 4-phosphate, which was a 4 carbon containing compound, it will combine with DHAP. Next, here we were having next 1 molecule of DHAP. It will combine with another molecule of DHAP, and in presence of the enzyme aldolase, it will be converted into pseudohaptulose 1,7-bisphosphate. Okay. Now it will be converted into pseudohaptulose 7-phosphate by removing 1 phosphate, and it needs the enzyme pseudohaptulose 1,7-bisphosphatase. Now we were having next 1 molecule of 3-PGL. This 3-PGL will be utilized over here with pseudohaptulose 7-phosphate. Pseudohaptulose 7-phosphate and 3-phosphoglyceric acid in presence of the enzyme, trans-ketolase will give ribose 5-phosphate and the xylulose 5-phosphate. Xylulose 5-phosphate, here we have one molecule of xylulose 5-phosphate, and in this step we were having one molecule of xylulose 5-phosphate. So we are having now two molecules of xylulose 5-phosphate in this cycle and one molecule of ribose 5-phosphate. This ribose 5-phosphate will be converted into its isomer form in presence of the enzyme isomerase. The isomer form of ribose 5-phosphate is ribulose 5-phosphate, and the xylulose 5-phosphate, two molecules of xylulose 5-phosphate will be converted into its epimer form. The epimer form of xylulose 5-phosphate is ribulose 5-phosphate. Okay. Now we are having ribulose 5-phosphate, three molecules of ribulose 5-phosphate. So we need to regenerate RUBP. That is the reason we have to add another phosphate into ribulose 5-phosphate. So we need a enzyme, that is ribulose 5-phosphate kinase, and three molecules of ATP are being utilized over here. So now we are done with this cycle. Thank you for watching this video till the end.